Alrighty, so I'm really excited to introduce my friend Gabe Rocklin, who's going to present our seminar tonight. So Gabe did his postdoctoral work at the Institute for Protein Design. He's rocking the t-shirt today, the IPD, doing de novo protein design and protein interface design. Gabe wants to understand why proteins fold and what makes them fold. And now is a faculty member at Northwestern. And yeah, Gabe, please take it away.
All right, give me one moment to share this and then rearrange everything so that, oh, can you, transfer to me? Or make me co-host or something. Yeah. Okay, let me do that, and then re-expand this so I can see the people that are willing. Yeah, cool. Well, I'm glad that I can see a few of you on the camera, so I can judge. It's really, really hard to give talks over Zoom because you can't tell how much people are getting. But from a few of you, I can tell if you're like nodding or really confused. But anyway, so because it's hard to do this without feedback, if you want to, please, you could turn on your mic and like stop me with questions in the middle if you want. Okay, so thanks, Chris, for introducing me. I started at Northwestern a year and a half ago. The lab has grown pretty fast. There's 8 people right now, and it might be 10 soon. And so I'll just give a really quick introduction to the lab before I talk about the work. We focus in a few different things. We try to focus on high throughput experiments and then interpreting all that experimental data using machine learning. So each of these different areas, the focus is on high throughput experiments that can teach us, not about one design, but about large numbers of designs and the properties that make them the way they are. So we try to design ultra stable protein scaffolds. We try to design, is everything? Yeah, okay. We try to design and discover proteins that bind to therapeutic targets. We try to discover, we're working to discover rules for delivering small proteins into the cell cytosol so that you can have therapeutics that interact with cytosolic targets. And then a big chunk of what we do is to understand the determinants of protein conformational dynamics. And that's actually what the second half of today's talk is going to be about. So I'm going to tell you two short stories that are both basically completely still work in progress. The first is about using a high throughput experiment to measure folding stability of designs. And this is where the why designs fail part of the talk comes in. I labeled this old work because it builds on work that I did a few years ago and has already been published, but there's actually like significantly new stuff in this area that I'm going to show. So it's like a big update of new stuff in that area. And a lot of that new work is actually being done by Hugh Haddix, who's a postdoc in David Baker's lab. And I've been kind of mentoring him on continuing that project. So a lot of what I'll show there is his work. And then the second part is going to be on the approach that we're taking to measure protein conformational dynamics in a high throughput way. And that's very much a work in progress, but I'll show you how the approach works and what we've done with it so far. Okay, so the question I asked a few years ago was, how can we improve our models of protein stability and design better proteins? And the motivation for this came because the significant fraction of most proteins that we designed when I was a postdoc in the Baker lab just didn't fold. They would be random coil or or they'd be molten globules, or even when things did fold, they frequently didn't function. And that was like in the projects that worked. Like you'd have a project that worked where half the proteins were successes and half the proteins were failures. And then there were other projects that just didn't work at all because none of the proteins were successes. So I wanted to understand what's going on. And the approach that I took was to design very, very simple proteins, very simple proteins on a large scale, like thousands at a time, and then screen them all for folding stability using a protease-based assay. And I apologize that some of you might be familiar with this work already, but it's just the background that I have to give that shows how the newer work has built on top of this. So to jump back a few years, I designed thousands of proteins in these 4 very simple topologies. And these are de novo designed folds, so we weren't copying the structure of any natural protein. And 2 different sequences in one of these folds might have a sequence identity of like 30 to 40%. So the sequences within the folds could be pretty diverse. And I designed thousands of these and then encoded all the designs on oligos that could be ordered as Agilent or Twist oligo libraries, and then used a high throughput assay to screen all those designs for folding stability. And I'll just quickly walk through how that works. We take the list of designs, every design sequence gets encoded on a DNA oligo, and then that oligo gets transformed into yeast so that every cell makes one different design. And we're using yeast surface display, which I'm sure most of you are very familiar with. There's a fusion protein construct where the test design is fused to AGA2, which gets displayed on the surface of yeast. And then there's a MIC tag at the end that can be labeled by a fluorescent antibody. So if the antibody labels the yeast, then that shows that the design successfully expressed and made it to the surface. And what we would do with these cultures of yeast is we would add protease. And if our test protein is very unstable, it gets cut very quickly by the protease. But if it's folded and stable, then it gets cut very slowly. So if we use flow cytometry to collect out the fluorescent yeast, then we collect the yeast to encode the stable designs, and we do deep sequencing to figure out what all those designs were. And the first time we did this, actually still the large majority of the designs failed, but about 20% in this one particular fold topology were stable. So that was about 200 designs, which was like way more than had been characterized in any previous like de novo protein design project. And from examining the properties of those 200, I was able to design completely new designs in all these 4 topologies and had much higher success rates. So eventually the success of these alpha, alpha, alpha designs went up to 85%. And what we learned in this fold was able to transfer over and get pretty stable designs in these other folds as well. And I'm not going to go into much detail on this because it's all old and published, but the main things we learned was that designs needed to be more hydrophobic, and they needed to have better agreement between the secondary structural preferences of amino acids and the secondary structures that those amino acids were in. You might notice that this particular fold was really challenging to design, and I didn't really have much success with it, but the first rotation student in our lab at Northwestern actually tried designing a 5th generation of this topology and was able to make about 2,000 new quite stable designs in that fold. And we're now characterizing some of these and trying to understand what's the difference between this topology and the others. Why did we fail at first, and what did we learn from this large number of new successes? Okay, so almost all of this is old and published. What's new, and the jumping off point for this to the project that Hugh is working on, is to try to understand, like, why didn't these success rates go to 100%? So in alpha, 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 even after we had all these examples of success and failure, like I really tried hard to make all the designs in round 4 stable, but a good like 15% were still unstable. And here, a good 50% were unstable. So what's going on? So what we started doing after this was to try to take large numbers of features of these designs and make regressions that would try to correlate the stabilities of the designs with the structural properties of those designs. And I'll show you some of those regressions here. So this is I think random forest regression that actually Francis Mata, who's a collaborator of the Baker Lab, he's not in the lab, just a collaborator, random forest regressions that were made from 113 different features. And each of these topologies, he made a different regression. And so that the features that go into this are like all the Rosetta score terms, as well as other things like unsatisfied hydrogen, like buried unsatisfied groups, or total hydrophobicity of the design. If you're familiar with Rosetta fragment quality, that's something that goes in. A list of about 100 things that were pretty simple to compute. And what you can see from these regressions is that they capture some amount of the variation in stability, but the regressions are far from perfect. And not only that, but there's some particular designs. There are outliers that can be quite far away from the fit line. So like if you focus on the HHH topology, these dark spots are these red spots are designs that are predicted to be very stable, but experimentally, they're basically like random coil. They're not stable at all. I'm not going to go into the details of what the numbers are, but you could ask if you're curious. But basically, just you can take my word that these designs are being predicted very poorly by the regression. They're way less stable than they should be according to all their features. And
Gabe, I have a quick question. Sorry to interrupt.
Yeah,
so
no,
with this this analysis here, on the x-axis you have predicted, and on the y-axis you have experimentally measured. Just out of curiosity, maybe you already mentioned it, but is there quite a bit of variance on, so given a dot, like if you were to do the experiment again, would the score be dramatically different at all?
No, the reproducibility from experiment to experiment is maybe like 0.2 or 0.3. So they're predicted to be 1.5, and experimentally they're 0. So it's far beyond the experimental variation of why they're so unstable. Mm-hmm. I should just tell you what these numbers mean. A score of 1 means that you're 10 times more stable than we would expect. You're 10 times more protease resistant than we would expect for a random sequence. And a score of 2 means you're 100 times more protease resistant than we would expect for a random sequence. And I should say that the protease stabilities that we're observing are not specific to any one protease. We do this with 2 different proteases that have completely different specificities, and we get basically the same results regardless of what protease is used. So we're not seeing protease specific things. We're seeing the effect of folding stability on protease resistance. So anyway, I'm highlighting these designs that are experimentally way less stable than they're predicted to be. And so what we hypothesized was that designs that are predicted to be very stable but are experimentally unstable, we hypothesized that these might be close in sequence space to stable designs. And also that if we study these designs, they might reveal stability determinants that are missing in the regression models. So, the regression models are saying that they should be stable, but they're not. So hopefully if we study them, we can figure out what the regression models are missing. And the other hypothesis that I'll show you in a second is we hope that these unstable designs were close in sequence space, and that we could actually use them. Specifically, we could do deep mutational scanning, and I'm going to show you that in a second, to try to find single mutants that would stabilize these designs and hopefully explain why they were experimentally unstable to begin with. Now, I just want to say, you might be thinking a few other ways that we could approach this. So like if our model based on 113 features is doing poorly, you could say like, well, why don't you add more features? And we did do that, and it helps to an extent. But there are difficulties there, and there's almost an infinite number of features that you might calculate. And the other thing that you might think is, well, why don't you just stop doing this manual featureization? Why don't you just do deep learning that automatically will figure out what all the features are? And that's a good idea too. the challenge there is that the training sets that you might train deep learning models on, like all the protein structures in the PDB, for example, well, there's a bias there that you're looking at stable folded proteins, and you're not necessarily looking at proteins that might be stable but are slightly off. And the other thing about that is that if you trained deep learning models to study natural proteins, then you might end up encoding features that vary a lot between natural proteins, but the differences between bad designs and natural proteins might be of a different kind than the variation that's found among natural stable proteins. So I don't think that approach, I'm not saying it won't work. I'm just saying it's not necessarily a sure winner, and it's not necessarily a replacement for having new experimental data. So anyway, like I said, we hypothesized that these designs would be close in sequence space to stable designs. So we used deep mutational scanning on these surprisingly unstable designs to find stabilizing mutants. And now I'm going to show you what we found. So this is a 3 helical design, and white. So the x-axis is every position in the design, and the y-axis is all the different amino acids that we could introduce at that position. And cells are blue, grid cells are blue if they're unstable, and they're white if they're stable, and they're red if they're very stable. So pretty much, like, this is different from a normal deep mutational landscape, because normally people do deep mutational scanning on functional proteins, and then they see what breaks that protein. We're doing the other thing, where we're taking an unfolded protein and seeing if there's any way to rescue it. And so what you see across this whole thing is that almost everything is blue, because most of these random mutants in this unfolded protein don't do anything to stabilize it. But there's a few mutants that are actually highly stabilizing, and I've circled some of the most interesting ones. So this is valine 11, and valine 11, the protein actually becomes much more stable if it's mutated to a smaller amino acid, to alanine, or even better, to glycine. So quite surprising, take a hydrophobic that's in the core and make it glycine, and you've actually stabilized the protein by a lot. And likewise, at this other position, at isoleucine 21, it also wants to be smaller amino acids. So you can mutate it to alanine, and that's really stabilizing, or you can mutate it to glycine again, and that's also somewhat stabilizing. And if you look at the designed structure of the protein, these two amino acids are actually directly in contact with each other. And if you evaluate the distances between the atoms, they're actually like a little bit closer than what's normally observed in crystal structures. So basically this Rosetta designed protein had a designed in clash that, according to the score function, was reasonable, you know, like, but is so bad, is so destabilizing that the entire protein is an unfolded random coil. And if you mutate one of these amino acids to alanine or glycine, then you relieve that clash and stabilize the design.
Gabe?
Yeah.
This is Andreas. Hi.
Hi.
So when you do the single point this bad mutation, did you do CD spectra on these? Is there some helical content in there still left, or is it just completely unfolded?
We have not done CD on the unfolded design. Hugh is, I believe, going to do that. I think some of the best evidence that it's totally random coil is that the entire mutational landscape is blank. Like, it's every point mutant is unfolded. So if there were tertiary interactions in the wild type protein, the original design, then I think we would see destabilizing mutants that affect that kind of weak structure. But instead, like, everything is totally protease sensitive, basically as protease sensitive as can be. So the fact that there's no destabilizing mutants is also strong evidence that the starting sequence is basically random coil.
Also really fascinating result. I'm just wondering, if you take that model and you put it through something like mole probity, does mole probity detect a clash?
That's a great question. Not being crystallographers, we did not think to do that, but that's a great thing to look at. So I'll say here that Hugh's done a lot of follow-up to understand, to observe that actually these distances in Rosetta design proteins were much closer than are observed in crystal structures. And if you take crystal structures and relax them, then things get closer than they were before. And he's done a lot of follow-up here. So he's working on an improved version of the energy function that will hopefully address this problem. And I showed you this in the context of one particular design, but we saw this pattern of large amino acids replaced by smalls being stabilizing across actually a number of these different proteins that we looked at. I should say we looked at 20 different of these proteins. I'll show one more example. So this is a...
Can I just ask a question there? Because I know from some of the other functional studies, we often see that we actually go the other way during the directed evolution from smaller to bigger. So what is it that we are somehow missing in the energy function, if you were going to speculate a bit on this?
Yeah. What's missing is that either the radii of these atoms were too small, so Rosetta thought that they could get very close to each other when they can't, or else the penalty of that clash was too minor. And Rosetta said, like, yeah, there's a little bit of a clash, but it's not enough to drastically I don't want to replace valine with glycine because I think the valine is still better, even though there's a little bit of a clash.
And maybe one more question, Gabe, if you don't mind.
Yeah.
I noticed that there was a leucine to glutamate mutation.
Yeah. Extremely curious, and I don't have an explanation, and we have not followed up on it, but I will tell you that this experimental data was actually done 4 times. So it was done in 2 independent experiments with trypsin and 2 independent experiments with chymotrypsin. So totally different specificity proteases in complete like biological replicates, going all the way back to like retransforming the yeast. And what I'm showing you here is the lowest stability of that mutant in any of those 4 replicates. So that bizarrely stabilizing leucine to glutamate is very reproducible, but I do not have an explanation of what's going on. And it's not a protease specific thing because it's stabilizing the protein in both the chymotrypsin and the trypsin conditions. Over here, there's lysine to leucine, and if you look at the structural model of the protein, this actually makes a lot more sense. But yeah, cannot explain that right now. So I'll show you one other example. So this is a mixed alpha beta protein, and what we found in this protein was 2 different mutants that actually do opposite things. So this is a very highly stabilizing methionine to valine mutant, so a large amino acid getting a little bit smaller, and this is a highly stabilizing alanine to methionine mutant, so small amino acid getting larger. And again, if you look at these 2 in the design structure, there's this basically cavity that both Rosetta and Pymol identify. It's not completely buried in the Pymol structure because you can see the surface of the cavity, but it is identified as a cavity with whatever probe radius, I guess probably like a 1.7 probe radius or something. So there's this cavity here, and these are the 2 amino acids that are highly stabilizing. So basically, if you make this methionine a valine, then you make it smaller, and this hairpin can move closer to the helix. Or likewise, if you make this alanine a methionine, then you kind of fill in that cavity, and now it's okay that this hairpin is far away from the helix. So basically, it's bad to have this mismatch of small here and large here, but if you make them both small, then the protein's way more stable, and if you make them both large, then the protein is also way more stable. So I think you could say that not penalizing this cavity could be, I don't know exactly how you would want to fix this in the energy function, but I think we were able to understand why the protein is destabilized by the mismatch in size and packing between these 2 amino acids, and you can kind of fix it from either angle.
So Gabe,
Yeah.
sorry, this is Jared. For both of these designs, if you actually mutate these in the model and then relax, does the structure seem more packed, better packed?
Yeah, I don't have the answer to that. But that's one of the things that Hugh is going to be doing. The first direction that he's gone with this was actually a big reparameterization of the energy function based on changing atomic radii to mainly fix the clashing problem. And then what he's just on the cusp, I think, of having an energy function that he's happy with, and then is going to take that, go back and relax all the models, see what they look like, and also see if when we make the regressions that I started with, can you more accurately predict stability score versus on the basis of structural features when you have structural features that are computed with a better biophysical model.
Cool.
Okay, so that's the end of the why were designs failing. I'll just summarize this by saying we examined all the point mutants in about 20 designs, and about half of them could be stabilized with just one mutant. So the designs that we picked genuinely were very close in sequence space to quite stable proteins, but had small flaws that were introduced by a imperfect biophysical model, and we were able to discover those through this high throughput experimental approach. Okay, so last for the second half, I'm going to talk about the approach that we're taking to understand protein conformational dynamics in a high throughput way. Okay, so most of you, all of you are familiar that proteins don't just sit in their folded structure. They always go back and forth between different conformations, and that includes conformations that are partially unfolded or fully unfolded. And so this is true not just for natural proteins, but it's true for even very simple designed proteins as well. So I'm showing you, this is one of my designs from a few years ago. It's 40 residues in length. We solved an NMR structure of it, and the NMR ensemble is very tightly converged to something that's basically the design model. But so this is what the protein looks like 99% of the time, which is what we're seeing by NMR. But if you want to know what happens 0.1% of the time, or 0.01% of the time, or 0.001% of the time, then you need to use other methods that can observe conformational fluctuations. And one of those methods is hydrogen exchange. So if you're not familiar with hydrogen exchange, you can take a protein that's in water and put that protein into D2O, and then you observe, so all of the backbone amides, the hydrogen will come off and it'll be replaced by a deuterium from the solvent. But that exchange can only happen if that part of the protein is locally unfolded, or if the whole protein unfolds. So basically, the rates of exchange at all the different positions they can tell you different things, but the way we do the experiment, it tells you the equilibrium between open, partially unfold, between open and unfolded and closed at that particular site. And what I'm doing here now is I've colored all the residues in this protein by those equilibrium constants, or the opening free energies. So red residues are folded almost all of the time, and blue residues exchange very quickly. And actually, so some of the blue residues, if a residue is on the surface of the protein, then it's possible that if it's not making a hydrogen bond with the backbone amide, it's possible to exchange even when it's still folded. So you just kind of ignore those. But for the folded positions, for the positions that are in secondary structure where the amide is making a hydrogen bond, that exchange rate, or that opening free energy, tells you the free energy of opening up the protein at that position. And I've put up the numbers here to show that these opening energies can vary by many orders of magnitude across even this very simple protein. So the opening energies in the red part are 7 to 7.5 kcal per mole. So these residues open up like a millionth of the time. And then you go down here just across this very short helix, and now the opening energies over here are 3 kcal per mole, or 3.5. So these residues open up 1,000th of the time. So there's 3 orders of magnitude difference in stability between the most stable residues in this helix over here, and these residues over here that are fluctuating much more frequently. And so we want to understand why is this? What is it about the structure and sequence of the protein that determine the local flexibility in all the different parts of the protein? Yeah, so the goal of this project in my lab is to be able to accurately predict these energy landscapes for any protein. So that means like, what's the overall stability and what are the energies of locally opening up all the different parts in that protein? And so our strategy for doing this is to devise a new high throughput assay to measure this. And I'm going to show you that in a moment. We're going to start by measuring very simple proteins and then work our way up in complexity. We're going to use designed proteins, and we're also going to intentionally design new proteins in order to sample the different things that influence stability. And then we're going to take the data from all these high throughput experiments and integrate it into machine learning models and also into physics-based force fields to be able to make accurate predictions on new proteins. So how does the high throughput experiment work? We start with the list of proteins that we want to examine. And then once again, we encode each protein in an oligo, and we order that as an oligo library. And then instead of expressing these proteins by yeast surface display, we express the whole library as a mixture in E. coli. So this grows in one E. coli culture. And every protein grows as a sumo fusion, thanks to Chris. And actually, you know, because the proteins are pretty small relative to the sumo domain, they all grow and express at relatively even amounts. So we can grow our 1,000 different proteins in one culture. And then everything has a his tag, so it all gets purified as one mixture. And then we cleave the his tag off. So now we've created this soluble mixture of designed proteins, or of a specified protein library according to whatever we put in the oligos. And then we take this mixture and we put it into hydrogen exchange mass spec. So what that means is that we take that mixture, we put it in D2O for 1 minute, and then we run that on mass spec, and that measures how much mass every different protein in the mixture has picked up. And then we also do that for 5 minutes and then put that into mass spec, and 30 minutes and put that into mass spec. And so each different time point gives data that looks like this. So this is the data for one protein, where that protein gets heavier at each different time point. But we're not just seeing this for one protein, we're seeing this for all the different proteins in the mixture. And so the proteins are resolved by liquid chromatography. They're resolved because they have different starting masses, and they're also resolved in a third dimension that's called ion mobility. And so those properties don't change. So even when the protein mass changes, you still know that it's the same protein because it's eluting at the same time. It has the same ion mobility signature. So you know it's the same protein. You can see how much mass it picks up at each different time point. And you can do this for 1,000 proteins at the same time. And I just want to say that if you've done hydrogen exchange experiments, you're probably familiar with experiments that break each protein up into peptides and then look at small fragments of each protein. We're not doing that right now in the data that I'm going to show you, but I think that we can adapt this method to do that, to get kind of local resolution of the amount of exchange in each different part of each different protein. We're not doing that right now, though. Right now, we're just looking at the amount of exchange in each intact protein. Okay, so this is where I start to really confuse people. So I'm going to walk through like the process in a lot of detail, just so that you get a good idea of how the data is generated and how we analyze it. So imagine that this is a protein, and every different residue exchanges at some different rate. But we're gonna walk through this just thinking about 3 different residues, like a very slow residue, a medium residue, and a fast residue. Okay, so exchange is stochastic. So even a residue that's in a very stable part of the protein, for some copies of, there's enormous numbers of copies of each protein in the solution. And sometimes that protein will just happen to be unfolded right when it was mixed into D2O, and that residue will exchange very fast. But on average, that residue is taking a long time to open up and exchange. So we get this first order exponential decay where initially all the copies of the protein have not exchanged, but after some time period, all the copies of the protein have exchanged at that residue. And so there's a first order rate constant that describes this exchange rate for one particular residue. And a medium residue is going to have a faster exchange rate. So it's going to, all the copies will exchange that residue more quickly. And then a very fast residue has a very fast rate constant. What we see by mass spec is time slices of this where we see the total number of exchanges at each time point. So at a very early time point, typically this protein among these three residues has picked up like one extra deuterium because the blue residue has usually exchanged. But sometimes it's picked up 2 extra deuteriums because the yellow residue has also sometimes exchanged. At a later time point, now typically there's at least 2 exchanges because the yellow and blue have probably exchanged. And sometimes there's 3 exchanges where the red residue has also exchanged. And then at a late time point, we usually see 3 exchanges, but maybe the red hasn't exchanged yet. So for sometimes there's only 2. But we're not just seeing this for 3 residues, we're seeing this for every residue in the protein. So there's a mix of first order rate constants for all the different residues in the protein. And we see these time slices that describe the complete distribution of added mass units at all these different time points. And then what we do to fit the data is we take these mass distributions, and then from these mass distributions, actually, you can very accurately determine what all these rate constants are for all these first order decays. And you cannot say which rate constant goes with what residue, but you can get the complete distribution of rate constants very accurately. And from those rate constants, you can convert that into the distribution of opening energies. So here I'm showing you the residues in this protein, but they're not ordered from the N terminus to the C terminus. So residue 1 is not the N terminus. This is just the residues ranked from most stable to least stable, where the most stable residue in this protein opens with an opening energy of a little higher than 4 kcal per mole. And there are about 5 or 6 residues that are like that. And then there's another set of residues that are around 3.5, and then so on. And you can see what the opening energies are for the complete distribution of residues in this protein. And I just want to repeat again, we don't know what opening energy goes with what residue, but what I'm going to show you in a minute is that I think we can learn quite a lot from these distributions of opening energies. And ultimately, when we make this experiment even better, I think we'll be able to experimentally localize these opening energies, but we're not there yet. Does anyone have any questions right here before I go on? Okay, I'll just add. Oh.
Hi, this is Nasos Ducis. Just how many time points do you need to be able to deconvolve this?
Yeah, that's a great question. We take 20 time points that can help resolve between like 0 and 4 kcal per mole. And then we take another 20 time points that kind of cover the range from like 4 to 8 kcal per mole. That's actually huge overkill. I haven't figured out exactly how little data you need, but we're basically just collecting way more data than we need because like the time requirements to do this and the sample requirements are very low. So basically it's all done using a robotic system, and every time point uses a tiny amount of sample, and it takes about a half hour of mass spec time per time point. So like 20 time points basically just run in a day. And it uses very little protein sample just because the mass spec's very sensitive. So if you want to answer that question, you could basically simulate it out and see how many time points you need. Yeah, we're doing a lot more than we need.
Out of curiosity, this is Eric.
Yeah, hi.
Since you're using ion mobility, what instrumentation are you using?
It's a water Synapt G2S.
Okay. Yeah.
Okay. So what can we? Oh, go ahead.
One more question, maybe.
Yeah.
So you showed, to explain the distribution of rate constants from fastest to slowest, you showed one structure. But then earlier you said there was like 1,000 different designs in there.
Right, so every design gets its own set of 40 rate constants, or however many residues it has.
In different folds.
Well, I mean, each different protein sequence is some different species that has its own mass distribution at each time point. So this is the mass distribution of this protein at the 15-minute time point. And then a different protein has a different mass distribution at the 15-minute time point. And we see that 15-minute mass distribution for all the proteins in the 15-minute run. And we see the 2-hour mass distributions for all the proteins in the 2-hour run.
But are all these different proteins, do they all have the same fold or different folds even?
I mean, in the data that I'm going to show you, they were all designed to have the same fold, but there's no limitation of the experimental assay that says they have to have the same fold. You could put anything you want into the experimental assay.
Okay.
Okay.
Gabe, can I ask one question to your free energy figure that you actually showed on the previous slide? So you started out by looking at these excited states of proteins. So would you expect that where you have a flat gradient on this landscape that you would actually have subpopulations during the folding?
Well, it's definitely true that the residues here that are opening up around 2 kcal per mole, they're doing some conformational fluctuation that changes the structure of that protein and happens like fairly frequently. And while these parts of the protein while some core set of residues is still staying folded. So some residues are locally opening up even when other residues in that protein are staying folded. Does that answer your question?
Yeah, it does. I was just wondering if you could go back then actually and expect what kind of time scale you would expect these hidden states, if I was going to use that expression for these subpopulations.
Yeah. So right now the experiments that we're doing are they're only giving us thermodynamics. They're telling us the populations of these excited states and not necessarily like the rates of going back and forth between open and closed. But you could actually do the experiments in a different way to try to see rates instead of populations.
Yeah. Okay, great. Thanks.
Okay. So there's not that many slides left. I just want to show like what we've concluded on this so far. So the question is, what can we learn from these energy landscapes on 1,000 proteins? And actually the data that I'm going to show you now is actually data that comes from just 400 different designs that were all designed to have the same fold. And it's actually this fold right here. It's a helix and a 4-stranded beta sheet. So it looks kind of like protein G, except smaller. So we're going to look at 400 designs that were all designed to have that same fold. And so these are those energy landscape curves, or those distributions of opening energies for 400 proteins that were designed to have the same fold. And I just want to add that even though we have not solved structures of all of them, we're quite confident that when if they're folded at all, they do fold into this structure. And that's because we've solved a number of structures and done a lot of deep mutational scanning studies and a lot of other control experiments that really give us confidence in those structures. And I'm going to illustrate that in a moment. But you'll just have to take my word on that, or we can talk about it more afterwards. So I've highlighted, there's 400 different lines here in gray, and I've highlighted 5 of them to show one of the most interesting properties of these energy landscapes. Actually, first I'll pull out this purple protein at the bottom. The purple protein was designed to fold into that structure, but as you can see, there's almost the opening energies are very close to 0, which means that this protein is basically unfolded. It doesn't really have much stable structure at all. So I wouldn't really say that the purple protein actually folds into the designed fold. But that's still interesting because there's something about that designed fold that's apparently unstable. Okay, but what's very interesting about these curves is that they sometimes cross each other. So the most stable residues in the blue protein are more stable than the most stable residues in the orange protein. Like these could be different parts of the protein, but whatever's the most stable in the blue is more stable than whatever's the most stable in the orange. But a large number of residues in the orange have kind of similar stabilities, whereas for this blue protein, after the most stable, like 15 or 16 residues, it suddenly drops off and the rest of the protein is much less stable. And we see a similar thing with the green and the red, where the red is quite stable across a large number of residues, but after the most stable, like 12 or 13, the other residues in the green protein are much less stable. So the way that I think about this is that the blue and the green protein are perhaps not very cooperative, because there's some significant chunk of that protein that can locally unfold even when the rest of the protein is staying folded. And that's very different from the orange protein, where basically everything opens up at about the same energy. So it's kind of like a much more all or nothing protein. We used NMR to characterize both of these proteins in detail. So this is actually, they were orange and blue on this previous slide, and now they've turned into blue and red. So what we did here, because our method can't identify the opening energies at each position, we used NMR to do that, and measured the opening energies at every site in these proteins. And what we saw was that in both proteins, the helix was the most stable part. And in this red, very cooperative protein, there's kind of similar opening energies, slightly less stable than the helix, across the beta sheet. But in this protein, this first hairpin is significantly more stable than its partner over here, than the counterpart in the other protein. But this second hairpin is very unstable. So the second hairpin can fluctuate off, and those hydrogens can exchange for deuterium very rapidly, compared with the other hairpin in the same protein, or compared with that hairpin in this different protein. So there's something about the structure of this that causes it to be much more dynamic and less cooperative than protein 1. And I don't know what that is yet, but I hope that by collecting enough data, we'll be able to, on a large variety of different proteins, we'll be able to understand why that is. And if you have any questions about this, I just want to show one more thing before I pause, which is that we also solved the structure of that non-cooperative protein by NMR, and we see that it folds exactly as designed, like 99% of the time. So this red part is the part of that very unstable hairpin. So like 99% of the time it's folded just as designed, but there are low energy fluctuations in this part of the protein that don't exist in that part of the protein, and also are not seen in like other different designs that have this same fold.
Could I ask a quick question?
Yeah.
Out of curiosity, have you thought about whether there might be intrinsic differences between designed and naturally occurring proteins? Whether there's bias towards more cooperativity and folding in naturally occurring proteins versus designs?
That's a really great question. And actually, just like last week, we collected this same data on all 200 proteins in the PDB that are under 50 residues and don't have any disulfide bonds. So we just measured HDX on all these small natural proteins, but we have not yet analyzed the data. So I basically, it's possible that we'll see much higher cooperativity in those proteins, but I don't know if we will yet. And I mean, to go like more theoretically, I think, yes, there's been lots of observations that natural proteins have been somewhat designed for cooperativity so that they don't sample like partially folded states that might be like more aggregation prone than something that's like completely unfolded.
At least it's part of, as far as I remember, textbook knowledge.
Yeah.
So it would be nice to have experimental data to actually confirm.
Yeah. The other challenge that we're going to run into is that right now we're defining like a cooperativity difference between two proteins in the same fold. Like this sequence in this fold is less cooperative than that sequence. But if we're just going to look at many different proteins and many different protein structures, then we need some kind of baseline of how much stability we would expect for that particular structure, as opposed to just compared to some other sequence that has a similar structure. So we're going to have
something else that may work is to look at things that are highly conserved, where you have a lot of sequences available and they're small enough to put it on a chip.
Yeah, so we also we have DNA, and we've basically cloned everything to start expressing like 1,000 different WW domains and 1,000 different LysM domains and 1,000 different SH3 domains, and it's all cloned and ready to express, but haven't started expressing it yet. Okay, so I showed you that like these energy landscapes are different, and in the last couple slides, I'm just going to show you a tiny bit about the very first baby steps in trying to understand those differences. And you know, like we want to do this on the 10,000 or 20,000 or 50,000 protein scale, where we have huge amounts of data and can try to see lots of things. But what I'm going to show you is just an incredibly crude and simple model to just look at these 400 proteins in the same fold. So the first thing we needed to do was to take these like complicated energy landscapes and simplify them down. And to do that, we used principal component analysis. So that's basically using dimensionality reduction to simplify down these landscapes down to just 2 dimensions. And so this is each line here is a protein, and now each point in this 2-dimensional space is a protein. And if I circle the colored proteins over here in this 2D landscape, then you can kind of understand what the principal components are. So the purple protein, the very unstable protein down here, is all the way on the left on principal component 1. And these highly stable red and green proteins are all the way on the right. So principal component 1 is kind of like overall stability. Now, principal component 2 is basically that cooperativity measure that I was telling you about. So the blue protein that has this big difference between the most stable part and the less stable part, that's all the way at the top on principal component 2. And the orange protein that's very cooperative and has very similar opening energies across the whole thing, that's all the way at the bottom. So I think of principal component 1 basically as kind of like an average stability, and principal component 2 as kind of like a cooperativity measure. And between these 2 principal components, you can see that they capture basically all the variation between these energy landscapes. So like 98% of the variation can be described with just these 2 principal components.
So Gabe, can I ask you a question? This is
Please.
David. Yeah, just wondering whether you have the protease assay also for these 400 variants, and whether that basically only correlates with the PC1 and there's no correlation with PC2, like how you just interpreted those.
Yeah, the old version of the protease assay actually doesn't correlate that well with this data, and that's because the dynamic range is actually pretty small, and even a lot of the proteins that are down here almost max out the stability range of the protease assay. We're now working with a better version of the protease assay that should hopefully have a larger dynamic range of stability, and it would both be able to like hopefully resolve the stability differences across here better, and also like separate like ultra stable proteins from medium stable proteins. I don't know if that's really going to work yet, but basically the answer is there's not that much of a correlation because a lot of this was basically above the high stability dynamic range of the protease assay so far.
Okay, yeah. Okay, cool. Thanks, yeah.
I will say that this assay is actually incredibly quantitative at measuring folding stability. Basically, if you look at the folding stability, if you look at the opening energies of the most stable residue in each protein, that's basically the folding stability of that protein in a 2-state model. And if we correlate that with what we see, not correlate, if we just plot that against what we see by like circular dichroism or guanidine melting, it's a perfect line with a slope of 1. And these numbers in replicates are reproducible to like a few tenths of a kcal. So it's a very accurate measure of folding stability here. Okay, so now this is the last slide. The question is second to last slide. The question is what correlates with principal component 1 and principal component 2. So we built a really simple linear regression that only has 6 features. And this is really just a demonstration of how you might go about looking at this. So this is 6 different features of these proteins. And what you can see is that with those 6 features, we can explain about a fifth or a quarter of the variation in principal component 1 and about a quarter of the variation in principal component 2, just with this really crude additive linear model with 6 features. And so what are those features? So what I've done here is I've plotted the coefficients of those features in the 2 different linear regressions. And the interesting thing, and why I'm showing this to you, is that different features of the proteins influence the energy landscapes in different ways. So the total number of nonpolar residues has a pretty high coefficient in the linear model of principal component 1. So it influences the overall stability by a lot, but it has minimal influence on the cooperativity principal component. And then a different feature like proline count that has a really high coefficient in the model, in the linear model of principal component 2. So it affects cooperativity by a lot, but it has a pretty small coefficient in the model of principal component 1. So these different aspects of the proteins are influencing their energy landscapes in different ways. And this is what I hope we can investigate on a much larger scale when we have more experimental data and when we start using more sophisticated machine learning compared with this really crude linear regression. Okay, so where is this going? We're going to collect much more data very efficiently. We're going to start studying larger proteins. We're going to change the experiments so that we can localize the opening energies rather than just doing this overall protein stuff. We're going to look at natural proteins and disease mutants, and we're going to start trying to improve the machine learning. And I think between all of this, that this will be a really exciting project. So last, I should thank a lot of people. Taeyoon Kim did the HEH design that I showed you. Sugi is a postdoc in the lab who's leading the hydrogen exchange project and will hopefully have really cool results over the next year. Since you asked about protease, I should say that Kotaro Tsubayama is developing the improved protease assay. Hugh Haddox did all the analysis of the rescue mutations. Francis built the linear regression, built the random forest regressions that we use to pick the proteins for the rescue mutants. Scott Hewliston solved the NMR structures and did the NMR hydrogen exchange, and the UW biofab collected all the protease stabilities of the rescue mutants. And give me 1 second before I come back and take questions. I'm going to turn off the beeping sound. Thanks. Thanks so much, and thanks, Chris, for the invitation, and thanks for all your great questions. And I'm happy to take any more questions that people have.
That's awesome, Gabe. Yeah, really exciting. I'm sure there'll be more questions. I'll take the privilege of the MC and ask the first one. Is actually, if you go back a couple slides there with the prolines in affecting cooperativity, that's really interesting. I know that Rosetta doesn't often put cis prolines in, but can. Just wondering if any of those prolines are cis.
None of the prolines are cis. It just seems like there's some effect of like really dampening local fluctuations in those areas. So, you know, the less cooperative proteins are the ones where some parts of the protein can kind of locally unfold, even if the rest of the protein is not. And I think that the prolines are really stopping that so that the whole protein's kind of like much more all or nothing.
Cool.
One question thought for your first part. One thing I've been always thinking, our high throughput methods generally become better and synthesis becomes cheaper and cheaper. If we were able to somehow, even if we can't solve it and make better designs, if we could point to those regions where we have bad designs, we could randomize them experimentally in initial library design.
Yeah.
I don't know if anyone is going in that direction.
Yeah, because I come from a modeling background, I'm always kind of interested in trying to take what we learn and put it back into improving computational modeling. But also, I'm really interested in using selections like that to create lots of data on basically what can be stable and what can't. So I this is almost going back to a 20-year-old experiment and now doing it with next-gen sequencing, but basically just taking designs and using degenerate nucleotides to make randomized cores and then measuring the stability of every possible core variant of that protein, and then seeing how well that, seeing number one, how well that correlates with modeling, and also number two, seeing how kind of the outer structure of the protein limits the size of what can be in the core, basically.
Yeah. And maybe even as simple as running more property over your designs and taking those clashes and just randomizing those positions.
Yeah, yeah.
Purely practical point of view.
I had a question that kind of relates to the last one. Really cool talk, by the way. I found this super exciting. I was just curious about the stage where you're designing the sequences for your fold of interest. And like, it kind of seems like you have this problem where you want to design as many as possible that seem like they're likely to fold to the correct structure, but also you want diversity so that you can like learn things.
Yeah.
And see what worked and what didn't. So how do you balance those two objectives?
That's a great question. I can tell you that it's not something that we've put enough thought into, and it's I expect that like as the models improve, then maybe the diversity will naturally go down because maybe everything will converge to like some best thing. There's the practical thing that we do is, number one, we use lots of different protein backbones. So even if the different backbones have the same overall fold, each backbone might prefer some different sequence, or they're different enough that you end up with reasonable sequence diversity. And then the other thing is when we end up with thousands or tens of thousands of things, we just cut out things that are too similar. So we manually filter the libraries to remove things that are too similar to each other. But it's not really an intentional strategy to create diversity. But I'm really interested in doing it in a more intentional way.
Hi, Gabe.
Hi, Una.
One, really good to see you. An excellent talk as always. This is also kind of related to those last two questions. Could you expand a little bit more on why, I believe it was like the HHH fold had so little success, and what was done in the new generation to improve the success of those designs?
Yeah.
And then like following up on that, or like kind of moving forward to combine the two aspects of your talk, I'm wondering how the hydrogen deuterium exchange dynamics principal component analysis could help with not just understanding how proteins fold, but like once you start engineering in like other functions into your protein, such as binding your other main goals, like how these will help inform those designs, or see how you can stabilize them but also insert function or something like that.
Yeah. Unfortunately, I'm not sure that I can answer either of your questions. So we have all these very stable HEH right now, and we definitely understand things within that fold that discriminated between stable and unstable. I don't yet have a good answer for like why it was actually that different from the other topologies, and that's kind of one of the things that Taeyun is trying to analyze right now. I think they're like, for one thing, like just to think about the beta sheet part of that protein, it's all edge strand. There's no central strands because it's just a hairpin. So there's no strand that's like hydrogen bonded on both sides. So it's kind of like the least stable bit of beta structure that you could have. And also I think there's something funny about one of the loop requirements, but I'm not really sure about that. So unfortunately, I will say that we saw cool trends within that topology, but I don't have a good answer yet for why it was that different from the other topologies. On how would you use the hydrogen exchange data to inform making more functional designs? That's a great question. I don't have a very good answer, except to say that one of the things we want to characterize is like many different variants of natural proteins that have function. Like I said, we're going to look at like 3,000 SH3 domains and 2,000 WW domains. And a lot of these are peptide binding domains. And we've thought about doing it like with and without peptide and see how the energy landscapes change. I don't know that really teaches you that much about how to design a better protein. I will think, okay, so I'll say that a common, hello, do you want to say hi to people, Natalie? I'm talking to people. Do you want to say hi? No, thanks. She says no, thanks. um, a common design goal is to have a design that's very rigid all around, because you want to present a thing that's not just globally stable, but it assumes a specific conformation and doesn't fluctuate away from that. And so I think that if we can understand the origins of these local fluctuations, then we'll be able to make designs that are much better at presenting specific pre-organized structures. And that's important, like for vaccines, that's important for binder design. A lot of things might not work very well because even though the protein folds into a stable fold, the key functional parts are not necessarily organized the way they're supposed to be. And hopefully collecting all this local data will help make sure the entire protein does what it's supposed to do.
I have a question. I was wondering if you anticipate that you could use your hydrogen deuterium exchange platform to, for example, design ligand binding sites or design proteins around ligands. Is that something that you would think would be feasible?
Well, the hydrogen exchange experiments could help like identify how the dynamics of the protein change when a ligand binds, or maybe help understand why some designs worked or some didn't. It's not really like a design method. It's like a experimental tool for examining conformational dynamics in proteins. But I think that hopefully it could, maybe that data could lead to new and creative design methods, but I don't know what those are right now.
Thank you.
Gabe, this is Nasos again. So I noticed on your helix, one of the examples, that it lacked prolines and aromatic residues. And I didn't know if that was maybe just an outlier, or if it was some function of kind of the overall design of those helix sequences. Were the loops, I don't know, you may have mentioned this earlier, but were the loops pretty short between the helices?
In all the designs so far that I've made, the loops have been short. They're like 3 to 4 residues. We definitely don't exclude aromatics or prolines. Lots of the designs have aromatics and prolines. I do think that as we try to systematically explore the origins of conformational dynamics, we want to intentionally make things that have longer loops, and I think that's going to be cool to explore.
Thanks.
I had a question about the first part. If you could help me think about that. So this blue rectangle with the few white and red spots in it.
Yeah.
That indicated, if I got that right, that you have an unstable protein, and there is just a few spots that actually help you fix it.
Yep.
Right. Now, if you reverse it, does that mean for that fold, I'm not sure this is true, so I'm phrasing it as a question, that you could destabilize that fold with just one residue, or did you have to,
Yeah.
I guess
So
the first question is, yes, if you pick the wrong spot, you can destabilize the whole thing. But is it true for the whole length of the protein? And that sort of reaches over into the second part where you have these cooperativity things.
Well, it's definitely possible in these mini proteins to find single mutants that, like, when you have a folded protein, we can find single mutants that are highly destabilizing. And we did a bunch of this deep mutational scanning in the published work and found like which sites are tolerant to substitution and which are not.
I see. So this sensitivity is not across the entire protein, just a few spots in there.
Well, usually it's like usually like you can't put polar residues into the core. It's kind of like what you would expect.
Yeah.
But you can look at that. Unfortunately, I have to go and help put kids to bed. But it was really nice getting to talk to all of you and have your questions. And if you want to talk more, then please send me email. I'm always excited to talk about this stuff. I'm sad that I couldn't be there in person and meet all of you.
Okay, thank you.
Yeah, thanks everyone.
Thank you.
When the pandemic is done, we'll have to have you come back. And
I would love to. All right, bye everyone.
Have a good night.
Yep.